Module 3. In this module, we will see the development of generic drug products. What are the generic drug products and how are they developed? Development of generic drug product by pharmaceutical industry is a scientific and technical approach which is totally different from developing a reference or innovator product. Most of the developing countries focus on developing the generic drug products because huge amount of investment is required for innovation and to develop reference product. The generic medicine has to be bioequivalent to the innovator drug and ensure the same biological effect with proper safety and efficacy. Nowadays, the pharmaceutical industries focus on the development of generic product as this does not require that much time and cost compared to the innovator company. But development of generic drug product is also difficult as it contains the same therapeutic efficacy as innovator. And usually the development approach is based on the target market, that is US market, EU market, etc. Developing a generic drug product in pharmaceutical industry is a scientific and technical approach which is totally different from developing an innovator product. It takes several years and stages to be registered as an innovator product. In terms of active ingredients, strength, dosage forms, method of administration, quality, safety, performance characteristics, and therapeutic indication, bioequivalence means that a generic drug product is virtually comparable to the originator product. Generic drug products are cost effective than their respective brand products. Pharmaceutical industries try to formulate and develop a generic product which is therapeutically equivalent to the innovator product. Though development of generic product does not require that much time and cost compared to the innovator company, but it is very difficult to develop a product containing the same therapeutic efficacy compared to innovator and to meet all the regulatory requirements as well. Let's see innovator and generic drug products. We have seen the generic drug products. What are the generic drug products? How about the innovator? Innovator products are also called as reference listed drug or patent drug. Innovator company or organization develop the medicinal product by trial and error and must have to conduct human trial or bioavailability study. And a generic drug is a medication created to be the same as an already marketed brand name drug in dosage form, safety, strength, route of administration, quality, performance characteristics, and intended use. These similarities help to demonstrate bioequivalence, which means that a generic medicine works in the same way and provides the same clinical benefit as that of the brand name medicine. There are some mandatory rules to develop a generic product for which few parameters are required to be same as the innovator for developing any generic. First is the active pharmaceutical ingredient and use. Second is dose and strength. And third is the route of administration. United States Food and Drug Administration, that is US FDA, clarifies some basic requirements for a generic product, which are generic products are the pharmaceutical equivalent of innovator product where active ingredients are present in same proportion, which we have seen in the earlier slides, in the same dosage forms and given in the same route of administration. We have seen all these three in the previous slide. Second is generic product is identical regarding biological activity where potential bioinvolution problems are not present. They are approved as safe, effective and have enough labels. They meet the same requirements for identity, strength, purity, and quality. They are made to the same exacting quality as innovator drug products as defined by the FDA's Good Manufacturing Practice rules. We know that the development of generic drug product in pharmaceutical industries require a lot of research and scientific review. And to meet the target launching time, proper strategic planning is essential. The formulation scientist has to know and analyze the detail about the product to be developed and the target market, as different market has different requirements. When a formulation scientist starts to develop a product for US, the product should comply to the physical and chemical parameter according to USP method, and it should pass the bioequivalence study against the innovator drug product of US. The product developed according to US guidelines cannot be marketed in EU or UK. Let's see what are the steps in the development of generic product in pharmaceutical industries. Let's have a brief about development. First is development strategies. What exactly it is? Generic medication makers, on the other hand, must show that their formulation is bioequivalent to the brand name equivalent in terms of quality and performance. When it comes to the excipient manufacturing processes and performance qualities, an innovative drug products formulation development has few restrictions. Then is patent restriction consideration. In this, the listing of relevant patents and duration of exclusivity for the approved drug product derives from the submission of an NDA with the FDA for a drug product developed with an NCE. The FDA's approved drug product with therapeutic equivalence evaluation, sometimes known as the Orange Book, contains this listing. Before the expiration of the patent, innovator holds the permission to manufacture for monopoly business. So before selecting the generic product to be developed, the industry should consider the patent status and market share for sure. Third is reference product selection and characterization. What exactly we do in this? We have seen that bioequivalence of generic product with reference listed product is the one of the prime criteria for development of generic product. Reference product selection for US market can be easily done by literature search at US FDA database and can be easily identified by their NDA number. What in case of EU market? In case of EU market, the formulation scientist has to review the public assessment report such as UK PAR or EPAR to identify the RLE. This PAR is nothing but public assessment report of the RLD. Again, EMA, which is European Medicinal Agency, helps on regarding issues as well. After having the RLD in pharmaceutical industries, the lab carries all the in vitro tests, especially assay, dissolution, and related substances. The prime target for a formulation scientist is to review the dissolution data of reference drug and design the generic qualitative and quantitative composition to match with dissolution profile, as percent predictability can be determined through in vitro in vivo correlation. Next is target market selection. How it is done? What we do in this? Actually, this target market selection determines and decides the way the company will develop the generic product, how to analyze the reference and generic product, the way stability test will be conducted, the way of selection of the batch size, and so many variables. If a company targets the US market, then all excipients should be USP grade. Analysis should be conducted by USP method, our in-house method and stability studies as well. Again, if a bioequivalence study is done against an European Union reference product, then is also acceptable for UK. In this case, only dissolution profile comparison should be demonstrating between the EU reference product and UK reference product. Then comes to manufacturing feasibility. What is this? It is always important to assess the manufacturing feasibility in pharmaceutical industries. Feasibility report characterizes the whole process of the development of generic product. To register a generic product for US, EU or other countries, the manufacturer must have a GMP or MHRA approved facility to manufacture the drug, to proceed stability test and for bulk manufacturing also. Then the auditor will visit the plant and as well as before shipment and prior to dossier and other paper submission. Next is formulation development. What do we do in this formulation development? First, we go for pre-formulation studies. Pre-formulation studies before starting the lab-based formulation, extensive documentation and studies carried out to assess the API. Even we monitor for specific molecule additional tests may need to carry. We do excipient selection during formulation development studies and designing of formulation method, selection of manufacturing method, then lead formula identification. All these activities are done in formulation development. Next is we do analytical method development, site transfer batch or submission batches. We manufacture the batches, we do the site transfer of these lab scale batches or the exhibit batches. Then we prepare a stability protocol, we keep our samples for stability study. Then simultaneously we perform the bioavailability and bioequivalence, that is BAB studies. So this is how we do the formulation development. Next is bioavailability and bioequivalence. What do we do in this? We know now we know that generic drug product has to be bioequivalent and interchangeable with the innovator or a reference drug product. Bioequivalence study can be done in fasting or fed condition. Sometimes both studies are required depending on the regulatory bodies. Sometimes we require fed and fasting both the studies. Sometimes as per the guidance we require only one kind of study. The European Medicines Agency, that is EMA, and FDA have released the guidance, that is the guidance for the bioavailability and bioequivalence. For solid dosage form and generic drug development, achieving the desired bioavailability is the major challenge. What do we see in this? Then the AUC between area under curve between the test drug and the reference drug should be not more than 90% confidence interval. Bioavailability can be in lower range even though where similarity factor is more than 50, because bioavailability depends on various factors like membrane permeability, human physiology, drug-food interaction, drug-drug interaction, etc. That's why bioequivalence study is so unpredictable. And bioequivalence is the evidence that a generic product is bioequivalent with the reference product. B study is more critical in case of drug with narrow therapeutic index. Next is fundamental assumption in bioequivalence study design. What do we do in this? A generic drug product that has been approved can be used in place of a reference drug product. A bioequivalence research is frequently designed after employing a crossover design to establish comparison within individual participants. Bioequivalence may then be assessed using reliable statistical methods while taking into account the pre-specified regulatory requirements for bioequivalence. Bioequivalence evaluation is one of the most contentious problems, as bioequivalence does not always indicate therapeutic equivalence, and approval of generic does not ensure bioequivalence. When evaluating bioequivalence for generic medication approval, there are four factors which are to be considered, main factors. First is, although the absorption characteristics of the drugs are not identical, they are therapeutically comparable. Next is, although the patterns of the drug absorption are comparable, they are not therapeutically identical. Third is, the absorption characteristics of generic and reference drugs are not the same and they have different equivalency. Fourth is, the last one is, despite the similarities in medication absorption patterns, they may not be therapeutically comparable. European medicines agencies consider generic medication products and RLDs having the same active ingredient to be bioequivalent if the rate and extent of absorption are assessed using plasma drug concentration time curves following administration in the same molar dose. Then there are regulatory aspects for the development of generic drug products. It is basically dependent on the territory. The regulatory standards of different nations throughout the world differ, making it extremely difficult to produce a single drug that can be presented for approval in all of them at the same time. What do we do in this? Regulatory agencies ensure the safety, efficacy, and quality of all the generic products as well as the production and distribution process. The regulatory authority's primary aim is to ensure that pharmaceutical goods are developed in accordance with the country's regulatory requirements. And the generic companies aim to meet those requirements. So, what do they do? We'll first see EU. In Europe, one of the most well-regarded and well-organized regulatory system in the world is EMA. EMA is responsible for the scientific evaluation of generic drug products developed by pharmaceutical industries, which is applicable for human and medicines agencies. In US, we submit ANDA, the abbreviated new drug application. It is the application for registering a generic drug in United States, which is one of the most demanding regulatory authorities in the world. Then what exactly is ANDA application? ANDA is abbreviated new drug application. This is the application submitted to the FDA for the approval of generic drug product and for marketing authorization. Though innovator product has submitted human trial study, but the generic industries need not to submit any clinical and animal studies to prove their safety and efficacy, as they will submit the bioequivalence study, which is compared to the innovator drug. It has created opportunities for the generic pharmaceuticals for developing and marketing generics within 180 days. Even though it took minimum 18 months for the final approval of ANDA by the FDA. Then, once we have submitted the ANDA, then it comes bioequivalence review process as per the regulatory guidelines, and finally the labeling part is reviewed. As we have seen in the previous slide about the development of generic drug product, let's see the formulation development procedure. This is a pictorial representation for ease of understanding. What all things we do in formulation development. First is pre-formulation study. We do pre-formulation study. Then excipient selection and designing of formulation method. Then selection of manufacturing method. The final formula identification. The analytical method development. And at last, we design a stability protocol. The regulatory standards of different nations throughout the world differ, making it extremely difficult to produce a single drug that can be presented for approval in all of them at the same time. Regulatory agencies ensure the safety, efficacy, and quality of all generic products as well as production and distribution processes. The regulatory authority's primary aim is to ensure that pharmaceutical goods are developed in accordance with the country's regulatory requirements, and generic companies aim to meet those requirements. So it is country specific. There are specific requirements for EU, USA, and ROW. That is rest of the world market. We have taken a small example of abbreviated new drug application which we filed it to US FDA. So what are the major factors or major parameters which are to be considered? First is types of certification. These are nothing but the patent certifications. Common technical document. This we'll see in other modules. The registration requirements of generic drug product in the US and ANDA regulatory review process. So all these things will go through in other modules. In this module, we have seen what are the generic drugs, what are the innovator drugs, development of the generic drug products, and what are the factors we consider during the whole development process. So this was all about module 3. As we know that a drug product is a combination of drug substance, that is API, active pharmaceutical ingredient, and excipients. So for registering the API, we submit the drug master file. So we will see what is the DMF and the types of DMF in the next module. Best luck.